Hello from Finland. It's such a pity that I couldn't join you on site because of the visa problem, but thanks to the organizing committee, I could still join there online. Today, I'm going to talk about my research on Phragmites. Genome-wide analysis checks the emergence of intraspecific polyploids in Phragmites australis. Phragmites australis is a widespread aquatic plant and is highly adaptive to different environments. It provides important component of a wetland system. It provides habitats for animals, but on the other hand, it becomes invasive once introduced into new territory. In United States, the invasive type of Phragmites was found to be in a small region on the east coast of United States. But after 50 years, it was found to conquer almost all over the country and forcing the Native American type of Phragmites to retreat to the north. The reason that caused the invasiveness is still unknown, but there are several things need to be noticed. These species have several ploidy levels, including 3, 4, 6, 7, 8, 10, 11, 12 sets of chromosomes with the haploid chromosome number to be 12. It can propagate both sexually and asexually with seed and rhizomes. A number of researchers have been trying to solve the phylogeny of Phragmites based on chloroplast and AFLP, as well as SSR genetic markers. Based on chloroplast, people classified the Phragmites into North American native lineage, North American invasive lineage, which was later found to be sourced from Europe, so part of European lineage, and the Mediterranean lineage, and East Asian and Australian lineage. However, these results are based on chloroplast. It's possible to bias the real phylogeny because of chloroplast capture. So nuclear genomes are needed to address the phylogeny and evolutionary history of the common reed. The most common seen ploidy level in this species is tetraploid and octoploid. Diploid was very rarely seen. Does it exist? Why it's so rarely seen? How did polyploidization occur and how does it increase the genetic diversity? How did reed become invasive? Our study will not address all these questions, but we will try to answer some of them. We sampled 88 individuals from all over the world and performed RAD-seq sequencing on them. These individuals included tetraploids, hexaploid, and octoploid. And then we selected 3 individuals representing 3 different geographic lineages and performed whole genome sequencing on them to assemble the reference genomes. We assembled these draft genomes, and all of the 3 genomes have good BOOSCO scores, means they are quite complete. So Y17 is the native North American lineage, has 922 megabase pair. Mediterranean lineage has a genome size of 840 megabase pair, and the Gulf Coast US land lineage have this 1.2 G base pair. To understand the evolution of the genome, we performed KS analysis by aligning the genome assemblies to an outgroup species, Oropetium thomim. And then we found for all these three genomes, there are 2 peaks. This suggests that the Phragmites have 2 subgenomes. So it it should be an allopolyploid. To understand the divergence of the subgenome, we took the both codes that are single copy in Oropatium and Oryza sativa in these outgroups species, but are duplicated in Phragmites. So this subgenome is diverged at 29 million years ago. To further confirm the genome assembly, the evolutionary history of the genome, we did another whole genome sequencing for a Chinese individual using PacBio Hi-Fi reads and high-C scan folding. We get a very beautiful chromosome level genome assembly with all the reads, most of the reads anchored to the 25 chromosomes. And the assembly yielded 870 megabase pair of reads. The genome, the Bosco score is 98.8%, so very complete. By aligning the chromosome level genome assembly to sorghum bicolor, we found that for each sorghum bicolor chromosome, there are two Phragmites chromosome matching it. So these two chromosomes may be the subgenomes. This is probably subgenome 1 and this subgenome 2. We did some initial phasing based on this synteny analysis and further phased it using a software called subphaser, which phased the genome based on the differential tamer in different subgenomes. So this software clearly separates the two subgenomes. Subgenome 1 is here and subgenome 2 is here. And now let's get back to the population genetics. Common reads are diverged into 7 lineages in the world, and so this is based on RAD-seq. And each lineage is characterized by its own ploidy level. As we have said before, using chloroplast, we were able to find five lineages, but using the nuclear genomes, we were able to find this North American lineage, 1, 2, 3, 4, 6 lineages. So Native American lineage, U.S. The land type lineage, and European lineage, Mediterranean lineage, the Chinese lineage, and Australian lineage. So these two lineages were separated by the nuclear genomes, and it could not, the chloroplast could not separate the European and CNN lineage. This is further supported by phylogeny, PCA, and fine red structure. So in this phylogeny and the population mixture picture, we could see that there are some genetic admixture. For example, this EU lineage is admixed with the MED lineage here and here, and then there's CN lineage is mixed with AU in this individual and here. And it's interesting that there are some population, some individuals have its genome admixture from 3 groups, and we will talk about that later. So to understand if ploidy level has played an important role in species in this intraspecific genetic variation, we performed RDA, redundancy analysis under constraint model using ploidy level as covariant. We also tried to use the genetic groups as covariance, and the results show the ploidy level explains 15.78% of the variance, and still more, not so much, but still more than latitude and longitude. They explain only 7.87% of the variance. Then we perform demographic analysis. So we obtain the derived allele frequency by aligning the RAD-seq to ancestral species. Then we perform stereo plot analysis. The stereo plot showed a sharp decrease in the population size of Native American lineage and land type lineage, which is expected, and then a mild decrease of the other lineages, and then we further we perform PSMC plot based on whole genome sequencing on the 3 individuals that we got, and then it shows yes, this North America and the land lineage had a sharp decline, but it also showed this Mediterranean lineage also had a decline. As we have explained before, there are a few lineages have genetic admixture of 3 lineages. In these individuals are in South Africa, and all the 3 lineages are tetraploid. So we expect this individual is also to be a tetraploid, but it turns out to be octoploid. All these 3 lineages, and also some other lineages in Romania. So this suggests that polyploidization can also be triggered by genomic conflict from different intraspecific lineages. So here is the take-home message. Phragmites australis is an allotetraploid, and polyploidization occurred at 29 million years ago. 7 lineages were detected in this species, and each species characterized by different ploidy level. Polyploidization may be triggered by gene flow between lineages. I have to thank my collaborators. Thank you.